In this video, we're going to discuss the components of a discussion section and what you need to do to write a good one. Our learning objectives are to develop, analyze, and critique discussion sections of lab reports. And so let's start with the basics. You need to include references. So this is really the first time in our writing process that we're going to need references, where we talked about the methods you might need a reference to the protocol you use or the lab report manual. But this is really where your references are going to come into play. You also are going to have probably this be your longest section in terms of writing. So you need to make sure you set aside enough time to really do this justice. It's written in a mix of past tense and present tense. You want to discuss your experiments that you conducted in the past tense. So you want to talk about your results, your figures using the past tense, because those are experiments you did in the semi-recent past. And you want to use present tense for known, established scientific facts. So if you're doing experiments about cells and you talk about the cell theory of biology or the organelles of a cell, that's all going to be in present tense because those are well-established scientific facts. The discussion we call a triangle shape or a pyramid shape. And you're going to start small. You're going to start with discussing your experiments and what you found. And then you're going to get bigger and talk about how it relates to the field. And then you're going to end with your biggest, and which is how it relates to the world and why it's important. So you start nitty-gritty and then you relate it to medium importance, and then you end with reaching out to why this is important for everybody. The discussion and the introduction are really where you focus on the story of your study. Writing a paper is not just saying, this is what happened, but instead you're telling a narrative. You're giving people a reason why they should read this paper, why your research is important, why they should fund your research. All of that needs to come through, mainly, again, in your discussion. You want to convince me that this is important, and you also want to convince me that you're capable, that you did these experiments well, and that we should trust the data and trust the results. So when you're writing your discussion section, you want to explain how your results relate to the goals of your study and whether it supports or rejects your hypothesis. You're going to reach back to that original goal and that original hypothesis. You want to interpret your results. So in the results section, you say, this is what happened. And in the discussion section, you put that into context. Why did that happen? You want to discuss any outliers or inconsistencies in your data. Why did you have that one value that's super, super high or super, super low? Or why is this trend the way it is? And then you want to place the results in the context of what we know about that organism or that system or what previous studies have found. In your introduction, you laid out a gap in knowledge. Why this is important, because we don't know the answer to this question, and it will help fill in this gap. In your discussion, you're going to do that. You're going to show, look, we knew this and we knew that, but we didn't know this middle section, this gap, and our results fill that gap. And you're going to place it in the context of what has previously been found. You want to state the conclusions and explain why your findings are important. They support previous findings. They're different from previous findings. And if so, why? What happened and why? You want to consider possible errors, and then you also want to reach out to future questions. And this, if you are a prolific science writer, this will kind of set the scenes for your next paper. So in the future, we're going to look at this thing, and hopefully you'll end up doing that. So a suggested outline is you want to restate the purpose of your experiments, and then recap the results and state whether it supports or rejects a hypothesis. And then you want to explain your interpretation of why the results occurred at a biological level. And then compare the results to your expectations, and then compare them to previous studies. So now we're building in the context. And again, we're starting small and we're getting bigger and bigger and bigger here. And then we're stating the major conclusions, the major takeaways. We're discussing concerns and potential issues, and then we're ending with a reach out to the future. Next steps, future steps. So again, in the beginning, when we say restate the purpose, we're going to open with 1 to 2 sentence overview of why the study was done, what's important, what's interesting about it. Then recap the results. What were the main results? Do they support the hypothesis? The summary should involve all of the different findings presented in the results. So you want to talk about all of the different graphs and tables. You're going to rehash the major trends. This group is higher than this group is higher than that group, and then objectively discuss outliers and inconsistencies. You want to explain your interpretation or why the results occurred. Why did these results happen? What's the science? What's the biological reason that you saw what you saw? Compare results to your expectations. Was it unexpected? Why? How would you explain that? Remember, results never prove a hypothesis. They either support or reject it. And negative results are okay. We didn't see any difference between these groups. Totally fine, not bad. You don't want to assign bad to your data. Data is neither good nor bad. It just is what it is. You want to compare it to previous studies. How do they align with that previous research? State the major conclusions. What do these results mean? This is providing that context. What are the most important takeaways? What do you want your reader to know when they walk away from your paper? And this should be broader than just your project. So you're fitting it into the field as a whole. You want to discuss concerns and potential issues of what might have gone wrong. Was it your fault? Was it my fault? Where was the issue? Was it sample size? Could you have done it with a larger sample size? Was it variation? The groups were just very different. Don't give me just a list of possible errors. You want to provide the context. And again, no data is bad data. We don't assign good or bad to our data. The data is what it is. And unexpected results do not mean your experiment failed. It's just that your hypothesis was not supported. And then you want to discuss the future steps. How should you follow up? Where should the next researcher start? In this video, we talked about discussion sections. So now it's your turn to write one.